Hello, my lovely anatomists and physiologists. Michele Glass here. We left off talking about the primary immune response being triggered by infection of one of your cells. Remember, the key feature there is ultimately your infected cells are destroyed. What we are ready to look at now is the primary immune response that's happening when you have that professional antigen presenting cell triggering the immune response. So we're looking at the sort of professional antigen presenting cell. So let's draw our cell, and this time I want to draw it sort of like a Pac-Man, because our antigen presenting cells are going to be phagocytes, which means it's their job, their profession, to go around, recognize cells that just don't belong, phagocytize them or engulf them, digest them, yeah, and then show that out. When I say show that out, I mean antigen presentation. So here, the very first step is, of course, let's say the antigen presenting cell finds and phagocytizes a foreign, let's just say pathogen. Remember, the antigen presenting cell itself is not part of our adaptive immune response. It's not having a receptor that recognizes name, rank, serial number for that particular pathogen. It's just like that very generalized. We talked about it previously as being a protein recognition receptor that's just kind of like, oh, you don't belong here. Gobbles it up, eats it, digests, and then we're ready to initiate our immune response, which is where the antigen presenting cell does antigen presentation. We said, remember, this was our very first step. And because this is a professional antigen presenter, is going to be using those class 2 MHC proteins. And so let's try to sort of draw that out. So we're again going to have like our flags, and let's just say make it triangular again, because we should remember, even though we're looking at these processes, these mechanisms separately, because we're learning them and that makes it easier to learn, that's not realistic to what's happening in your body. If you have infected cells, you also have phagocytes that have found that pathogen. And so you would expect both of these mechanisms that we're talking about here to be happening. If one is happening, the other is happening. That's how you should consider this. And so we see our antigen presentation. So as we're thinking about our steps, the next basic step in our pattern was for an inactive lymphocyte to recognize and bind. And in this case, because we're looking at, oops, sorry about that. Because we're looking at the class 2 MHC proteins, we're going to get the CD4 T cell to recognize and bind. So now let's draw in our CD4 T cell. And I want to give myself quite a bit of room. So I'm going to put my CD4 T cell up here. I'm just going to make it cute, give it a little face. Nice little smiley. The CD4 T cell is the helper. it's going to give us a helper cell. If I can move it. Well, I'll stop playing around with that, but that's pretty cute. So I'm going to leave it. So the CD4 T cell will recognize and bind. Let me label this as our CD4 T cell. And I kind of got it too far away. So let me adjust my drawing just a little bit so that I have sort of an antigen that's presented a little bit closer to where my drawing is. These are specific, remember? So the antigen presenting cell was not specific. It was not one of our adaptive immune responses. It's our first line of cellular defense. It's a phagocyte, so it's one of our innate defenses. But the CD4 T cell is a lymphocyte. So now we're bringing in that specificity that we see here with our adaptive immune response. And now we're ready to go back to our steps. So we have a pattern of once we have recognition and binding, we get the co-stimulation. Remember, this is our go ahead. And in this case, it's the phagocyte that says, yep, we're good to go. So I'm just going to draw an arrow from the phagocyte to my CD4 T cell. And I'm just going to say, go ahead. Okay. And then the fourth step is for the CD4 T cell to activate. And anytime we're talking about a lymphocyte activating, we mean divide and differentiate. Divide is, of course, make more cells. Differentiate means they can have different functions. And so what we see being formed in this case, we get memory cells. I'm going to just write the word memory and stop because I'm going to add more detail. The CD4 T cell, when it divides and differentiates, makes the memory cell and it makes a helper T cell. And you can abbreviate this with a capital T and a lowercase h. And what we'll actually see is there's two types of helper T cells. So now I'm going to use my abbreviation, big T, little h1. That's a helper 1 T cell. These are going to release cytokines to help regulate macrophage. Remember, macrophage is going to be one of our professional APC cells and other T cells. So that could be your cytotoxic T cell we just talked about. And then we get another helper T cell. So I'm going to abbreviate this, big T, little h, number 2. The helper 2 T cell is going to act on B cells. And I'm going to write B cell in a different color so we can pick up on that in a minute. So when I go back up to my memory, we're making memory helper T cells. So I'm going to do my capital T, little h for abbreviation. And this is the same significance that we had before. Anytime you see the memory cell, it's not active in the primary immune response, but it's active in the secondary immune response, meaning all subsequent exposures. And it speeds the whole process up. You don't have to go through all these steps to generate a helper T cell. Instead, they're there, they're available, and they become active as soon as that pathogen is encountered. So it makes this whole process super fast. Okay. What we're going to see happening next. So let me actually draw from my CD4 T cell. Let me draw a helper T. So here I'm just going to draw another circle and I'm just going to label it T H2. Okay, so I didn't give it a little face. I still want to give it a little face, so I'm gonna kind of make it look like it's the profile. Okay. And what we're going to be looking at next is what's happening with the B cell. Okay. So it's sort of like a separate process, but related. So it's sort of like a separate process, but related. So I'm going to go ahead and number it, and I'm going to start again with number 1. I'm going to go ahead and number it, but I'm going to start again with number 1. So we have an inactive B cell that does antigen presentation, and it's using MHC2 proteins, and at the same time it's gonna have these IgD antibodies. Okay, so just to the side, let's say an antibody can also be called an immunoglobulin. And we can abbreviate that capital I, lowercase g. And there are, I think 5 classes. There's 5 classes of IgG, 5 classes of Ig, 5 classes of immunoglobulin, 5 classes of antibodies. And we're going to talk about each of those. We'll have a whole video just on the antibody that's coming up next. When we look at a B cell, so I'm going to draw my B cell just down here. So I'm going to give it a B cell. Oh, you know what I'm going to do is I'm going to use my letter B to be like the eyeballs. Looks weird. And smiley face. I like this one. I like this one. Okay. It's going to have IgD antibody hanging off. Antibodies are usually like a Y shape, so I'm drawing a bunch of little Ys. And because we're having our antigen marker be like triangular in shape, I'm going to show like a triangle bound. And I don't have to show that triangle on everyone. That's time consuming. And it's doing antigen presentation as well, which means it has also engulfed the pathogen. It's also engulfed the pathogen. So let me do a flag. Let me do a flag, the triangular flag hanging off as well. We would classify this as a sensitized B cell. Let me highlight sensitized B cell. That's what I'm talking about here. So I'm going to actually highlight this whole first step. This is how we know that the B cell is sensitized, because it's doing antigen presentation and the IgD antibody is bound to antigen. So it's kind of like a double display. Let's think of it that way. It's like a double display. But this is actually like that first step. We're doing antigen presentation. We just have this additional display. Remember that our next step is recognition and binding. And so what we see here is the helper 2 T cell is going to do the recognize and bind. So I'm going to try to move my B cell, or copy my B cell, move it up to the top. Okay, so I moved my picture of my sensitized B cell up to the top of the screen, where my picture of my helper T cells located. And we're ready for our step 2, which is that helper T cell is going to recognize and bind. And so let's show that happening here. This is the same sort of idea where we have that specificity of shape. And then we're ready for step 3, which is the same as what we always have. It's called co-stimulation. But this time what we're going to see is that helper T cell is going to release cytokines. So this time it's just we know more information about what that co-stimulation looks like. And so we can draw that sort of co-stimulation. So let's do, I'm going to make it look like dots of cytokine to the B cell. I'm going to label those cytokines. Okay. And then let's be ready for step 4. It's the same step we always have. So now we see that the sensitized B cell activates. And when lymphocytes activate, what that means is divide and differentiate. And in this case, we're making a memory B cell. Oh my gosh, you guys know the story. Memory B cell is not active in this primary response, but it will be active in the secondary, and it will be available super duper fast, ready to go with that next exposure. And then we're also going to be making a cell here called the plasma cell. And our OpenStax text actually lists a plasma cell as a 4th type of lymphocyte. You might want to do the same thing, but here's the key thing. I'm highlighting the plasma cell. I'm gonna put a box around the plasma cell. This is the cell that's going to secrete antibodies, or we can call them immunoglobulins. We're going to see first the secretion of IgM. So that's happening first, and we're going to talk more about this in the next video. And then we see the production of the IgG coming second, and this is the most effective at really getting that pathogen out of the body, and we're going to talk more about that in the next video. So back up here at my picture I've drawn, I want to draw my plasma cell. I don't know what I want it to look like. I'm giving it big, cute eyes here, big smile. And then I think I'm just gonna show Y-shaped antibodies coming off of it in like all directions. So the antibodies are secreted into your body fluids. So that's why we talk about it as the humoral response, where humoral is referring to your body fluids. And so it's the plasma cell that's really releasing those antibodies in your immune response here. But notice that to get here, to get to the plasma cell, you did have to have that helper T cell. You did have to have that helper T cell. All right, up next, let's investigate those antibodies in more detail. And as always, take care of yourselves and each other.